And we certainly feel like old friends these days, I tell you. So thank you very much for inviting me to speak at this fantastic meeting. What I'm going to do this morning in my talk is walk you through the history of cellular therapies for Parkinson's disease. So I'm going to go through a little bit about what the past has told us, where we are at the present time, where we're going into the future, where we would like to go in the future, and where we wouldn't like to go with some of the therapies which are currently out there. So to begin with, I thought it was important to emphasize what it is that we are trying to achieve with cell-based therapies for Parkinson's disease. And these cell-based therapies are essentially around dopamine nerve cells. So right from the outset, this therapeutic approach is not a cure. It will never cure anyone with Parkinson's disease because it's not dealing with the underlying problem, and it's not dealing with problems outside of the dopamine system. So then the question arises, well, why bother doing dopamine cell-based therapies at all? And the rationale for doing this is quite simple. We know that despite the pathology being at many other sites in the brain and possibly outside the brain, central to Parkinson's disease is the loss of a population of cells at the top of the stem of the brain, the dopamine cells that live in the substantia nigra. And you have 500,000 dopamine cells in each nigra, and when you lose half of those, 250,000, you get the first clinical features, motor features of Parkinson's disease. So that's what we're trying to replace. So that is a tractable problem, given that your brain contains somewhere between 70 and 100 billion nerve cells. That's a number we can cope with. Now, why would it work? Well, it would work because we know when we give people dopamine drugs, they get a fantastic response. So many people in this auditorium will know when they started their journey with Parkinson's disease and were put on dopamine drugs, the response was pretty spectacular. So we know that dopamine replacement therapy with drugs works very well in early stage Parkinson's disease. The problem is it creates problems in the long term. So our dopamine cell-based therapy is designed to do as good and as well as the drug therapies, but it will do no better. But if we can emulate that, and we can do that over time, then that will be a major advance in the natural history of treated Parkinson's disease. And the reason one gets complications with oral drugs for Parkinson's disease is twofold, really. One is that the dopamine drugs you take don't just stimulate the dopamine areas where you are deficient with the Parkinson's disease, but it stimulates sites where dopamine works normally and the system is normal. So you get problems related to overstimulation in sites of the brain where you want the drug not really to be working. And secondly, when you take the oral dopamine drugs, they don't simulate what happens when dopamine is normally active in the brain, i.e. it's non-physiological. And so that off-target effect and that non-physiological delivery of oral drugs sets in chain a series of problems which lead to the long-term complications that you see. So in theory, a cell-based therapy for Parkinson's disease, which is simply replacing the dopamine cells, will give you a response as good as your dopamine drugs without the long-term complications whilst not curing you. And I'm going to go on to explain exactly how that has panned out over time. So if you are thinking of doing this, and I'm just going to give you a little bit here on the sort of structure of how we try and take a cell-based therapy to the clinic, this is what happens. You essentially identify your cell source, and I will come back at the end to talk about the stem cells we're thinking about for dopamine cells. You then test it in an animal model of Parkinson's disease, and you would have heard a lot about that over the last few days, that there are no good animal models of Parkinson's disease. But for our studies, we don't need an animal model of Parkinson's disease. We simply need an animal model that loses dopamine, because that's what we're trying to do. Now, if we identify our cell source, transplant it into an animal model of dopamine loss, mimicking the problems you see in Parkinson's disease, and you get a positive response, you can then take that forward. You do a little open label study, open label meaning that everybody knows they're getting the treatment. So I know I'm giving you the active treatment, you know you're getting the active treatment. If that works, we can then see, are we doing it optimally? We then optimize what we're doing, and when we think we've got the best possible way of doing it, we then subject it to the gold standard of therapeutic trials, which is this double-blind sham surgery trial, where neither the physician nor the patient knows whether they've actually had the therapy or not. So that's the format by which you take a cell forward from the lab into the clinic. And this has been going on for about 30 odd years, and lots of different cells have been tried. But the only one which currently has stood the test of time are the fetal dopamine cells. So these are the developing dopamine cells in the fetal midbrain. And in animal models, if you take those out, so you take them out of the rat developing midbrain, transplant them into our standard model of Parkinson's disease, which is a dopamine deficient state on one side of the brain, we find that these dopamine cells can survive long term. They connect with the rat brain around it, the rat brain connects with it, they release dopamine, and animals get better. And that was done through a series of pioneering studies in the 1980s in Lund, led by Anders Björklund, in which our co-chair Patrick Brundin was very instrumental in taking forward through those preclinical steps. So by the mid-1980s, it was established that this technique, which could be reproduced in many labs, showed that if you took tissue from the same species, which in our animal models are rat to rat, mouse to mouse, but obviously in the clinic would be human to human, we took it at the age when the dopamine cells were normally developing, which in the human developing fetus is 6 to 8 weeks of age, and you put it where dopamine works, namely in the striatum, then it works very well in preclinical models. So what happened when it went to the clinic in the late 1980s? So the very first studies which were done of significance were done in Lund, where Patrick was heavily involved, and this involved taking patients and putting the tissue in open label studies. They knew it, everyone knew they were getting the transplanted tissue. And patient number 4, who I'll come back to a bit later on, was very important patient. So this patient in the late 1980s had a transplant of human fetal dopamine cells into one side of their brain, and then over a 3-year period, which is hard to see on this particular slide, they actually improved from being off 2/3 of the day with optimal drug therapies as existed in the late 1980s, and 3 years after having a transplant on one side of the brain, they had gone from being off for 2/3 of the day to being on all day. That clinical improvement was matched by imaging improvement. So this was an F-Dopa scan done at the Hammersmith Hospital in London, which looks at dopamine levels in the brain. And on the grafted side, you can see that there was a restoration of dopamine at the site where the cells were implanted. So the patient improved, their scan improved. On the non-grafted side, you can see that the disease process carried on. So clinically they improved, there was imaging evidence for it improved. Similar studies done in the US demonstrated that when one of the patients died from an unrelated cause 18 months after receiving such a transplant, the clinical improvement, the imaging improvement, was matched by the histology, showing that there were dopamine cells at the site of transplantation. So they work clinically, they survive, and then in a study done in the late 1990s at the Hammersmith, they also demonstrated that the transplants could release dopamine in a normal physiological way. So this was all very encouraging. The problem that then complicated the field in some ways was that in the early 1990s, 2 studies were funded here in the United States to look at this in more detail using these fetal transplants in these double-blind placebo-controlled trials. So these were where patients either had a transplant or were pretended to have a transplant and then were followed up to see whether it made any difference. And this was the headline in the United Kingdom in 2001 when the first of these was published. It is very typical of the newspapers, this headline. As I've said many times before, there are only ever 2 outcomes from any medical or surgical intervention. It's either miraculous or it's catastrophic. There's nothing in between, because that's what sells you newspapers. So this miracle cure that I've told you about has not just not worked, it's become catastrophic. So what was the catastrophe? Well, these are the two papers which were published in 2001 and 2003. The first of these papers showed that at the end of 1 year, when the patients were asked whether they were better for a transplant, the answer was no. And some of the patients had now developed a side effect that hadn't been seen before, was that the transplant itself seemed to be driving some of the dyskinesias in the patients. Of course, with L-DOPA-induced dyskinesias, you can always get rid of them, because you can always get rid of L-DOPA. If you have graft-induced dyskinesias, you can't get rid of the transplant. So this was a concern, but there were issues about exactly how that tissue had been implanted. And then in the second study, published 2 years later, there were similar results. There was no significant benefit comparing the transplanted patients versus the non-transplanted, and side effects were seen again. So now the field is sort of saying, well, it doesn't really work, it creates side effects. And then a few years after these two papers were published, there was these two reports followed by a subsequent report showing that the transplants themselves had acquired some of the pathology of Parkinson's disease. Not a lot of the cells, but some of the cells. So this implies that this whole approach is doomed. It doesn't work, it creates side effects, and the cells you're putting in develop the pathology which you're trying to treat in the first place. The problem is there is discordance with this. So this is a paper which appeared earlier this year, which was patient number 4, who I showed you earlier on, 3 years back to near normality with their Parkinson's disease, a scan that had returned back to normal, and they died in 2013, 24 years after they had their transplant, and at post-mortem, they had a transplant that was alive and healthy. So this is the area of the transplant, and you can see that throughout the area where the graft is placed, there are surviving cells and fibers innervating the brain. Now, there is pathology in that transplant, but the majority of the cells have survived and look healthy. So anatomically, these grafts can survive long-term, and there's also evidence of long-term graft survival clinically. So this is one of the patients who was grafted in Sweden, who's looked after by my good friend Tom Faltney in London. This gentleman is 20 years after a transplant, 30 years into his Parkinson's disease. Those leg movements are his graft-induced dyskinesias, and he's on no medication. So when it works extremely well. So in these two patients who were followed up in London, they not only returned to a motor score which was better than when they presented 30 years previously, but the dopamine levels in their brain were normal. So this is a little bit perplexing because we now have a problem. We have one set of studies that tell us it doesn't work and you get side effects. You have these anecdotal patients who seem to have done extremely well. What do we do? How do we move forward? Do we just sort of scratch our heads and say, well, it's interesting, let's just leave it and try something else? So one of the things to do is to go back and look at the trials and say, well, what is it about the trials that may give us clues as to why these different studies produce different results? And if you compare them, and you don't need to see the detail of this, you start to realize it becomes incredibly difficult to make any firm conclusions. And that's because all of the trials have used slightly different types of patients, different age, different stage. They have used different doses of cells. Some have used very few fetuses, some have used a lot of fetuses. The way in which the cells have been delivered with different instruments by different routes. Some of the patients received no anti-rejection immunotherapy after the transplants, some have received it more long-term. All of the studies have used different endpoints. When do you think the transplants had the most effect? 1, 2, 3, 5 years. They've all used different endpoints, and they followed up patients differently. So it's very easy to come to premature conclusions if everything is so variable in this way. So the analogy I often say is that in 1953, if I had taken 50 patients with a chest infection, given them 400 milligrams of penicillin, and done a chest x-ray 5 days later, I would have discovered that penicillin does not help chest infections. And the reason for that is that all the patients have, many of the patients will have different chest infections. I've only chosen one dose instead of getting the right dose. I haven't waited long enough, and I've used the wrong primary endpoint. And if I had followed that to its logical conclusion, I would have said penicillin doesn't help chest infections, therefore antibiotics don't help kill microbes. Let's abandon antibiotics and go back to putting people up in the mountains for fresh air to treat their infections. So it's very easy to arrive at a premature conclusion. It doesn't mean it's wrong, but you have to argue with the evidence you've got as to whether this has any merit. So what do we do next with this? So we went back and analysed the data in order to decide whether we could do a new trial. So we went back and put all the data together from the trials and discovered that those patients who did best were younger with less advanced disease. We also tried to understand why some of these patients had developed these graft-induced dyskinesias, and what we discovered was that it related in part to how the transplant was put into the striatum, whether it evenly distributed across and innervated it normally, and whether there were contaminating nerve cells which developed next to the dopamine cells, 5-HT neurons, which release dopamine in a somewhat indiscriminate fashion. So based on that analysis of pre-existing data, we decided to move forward into a new clinical trial. And that new clinical trial is TransEuro. It was funded by the European Union and is still ongoing. And what this trial has tried to do is it's tried to recruit the right group of patients that we identified by looking back on the data. It's been tried to improve how we deliver the tissue, so we put in enough dopamine cells, but none of these or little of these 5-HT nerve cells, and we deliver it such that it evenly innervates the area where we've grafted it. We will then take some of the patients who we are following up longitudinally in this study, transplant them, and then see them at 3 years to see what effect this transplant has had. There's other nuances to this about trying to blind the investigator and such like on the assessment, and we have currently grafted 8 patients using this fetal dopamine tissue. Now, the problem with fetal tissue, outside of the ethical concerns that it raises, is there are major logistical problems with it, getting enough tissue, and ultimately you cannot control what you're doing because every single patient will have a unique transplant from their own unique collection of aborted fetal tissue. So this has always been seen, this trial, as a stepping stone to the future, which are with stem cell-based therapies. So why are we interested in stem cell-based therapies, and which ones are we particularly interested in? Well, I've already briefly mentioned about the logistical advantages. Depending on the type of stem cell you use, there are there are less contentious ethical issues, but they're not insignificant with many of them. But you can actually much more define what you're going to produce. So you can, at the end of the day, say, I know what is in my product because I've engineered it to make exactly what I want. Now, as you are aware, there are many, many types of stem cells out there, and stem cell therapies have been in the clinic for decades in the world of blood disorders. Lots of these, frankly, for the use of neurological disease, are a nonsense. So there are lots of therapies that are out there that you can buy which have no scientific merit. And the only cells which I would say currently have any mileage in terms of helping people with Parkinson's disease are embryonic stem cells, so the cells which are made from spare embryos, or induced pluripotent stem cells, which Marlin will be talking about after me, and Steve after that, which I'm not actually going to say anything more about. Now, why are we interested in using embryonic stem cells? Well, the reason for this is that in the last 5 years, there's been a revolution in the science of understanding how dopamine cells normally develop, which we can then exploit to make embryonic stem cells into dopamine cells. So this paper from the Studer group in New York showed how you could make what looked like pretty good dopamine cells of the type you wanted for Parkinson's from embryonic stem cells. And then Marlin Palmer and her team have worked on a similar protocol where they have now demonstrated that they can take human embryonic stem cells and they can turn them into the right type of dopamine cell which is needed to repair the brain in Parkinson's disease. Because you can't use any dopamine cell, you have to use a nigral dopamine cell. That's what you have to produce. And the protocols are there now that you can make authentic dopamine cells that look just like the fetal dopamine cells we've used in clinical trials in the past, and they work with a similar potency and a similar efficacy. So we're now on the cusp of thinking, how can we take this forward into clinical trials? Now, learning from the past, what we've tried to do is to protect ourselves against the premature use of these at times before we really know what we're doing, even though when we do go to the clinic, it is going to take some time before we've optimized how best to use them. So we have set up across the globe an initiative called GForce PD, which is designed to bring the major groups together who work on dopamine-based therapies from stem cells for Parkinson's disease. So this includes groups in North America, groups in Europe, and groups in Japan. And we meet once a year to discuss where the data's got to, how we can take it from where we are now into a clinical trial. And as it stands at the moment, the group in Japan are thinking of doing an induced pluripotent stem cell transplant trial next year, although that may move back a year. The group in New York, led by Lorenz Studer, are planning to do a trial in 2018, and ourselves, we're planning to move forward in the next few years to doing a clinical trial. Now, in our particular case, what we're going to do is we're taking the fetal transplant trial that we're already running. The patients who are recruited to that will feed in to our new trial using stem cell-derived dopamine cells. That is currently in the final stage of preclinical testing because the regulatory authorities have very strict guidelines about how you test these cells to make sure they're safe. So we're currently seeking funding to do this. Now, this is not cheap, so the grant we currently are putting in to a UK charity is for £2.6 million, which is about the same as dollars and euros these days, £2.6 million to basically produce a cell and test it. That is what it costs just to make the cell, to make sure it's of the quality you need. There is no science in that, it's simply production and testing. And the hope is that that will take place in the next 2 years, moving to a trial which we will do in 2018 and 19, which will be an open label study, because it has to be when you're doing a first in human, and you're just going to test whether this is safe, with a hope that you'll get some signal of efficacy, and then work out how to optimally deliver it. Now, we are not the only people in this area. This is something which I'm sure a lot of you have seen, which is a trial that's taking place in Melbourne in Australia at this time. So this is the first stem cell trial in Parkinson's disease, which has been approved, if you like, by regulatory authorities in Australia. This is run by International Stem Cell Corporation, and many of you, as I say, will be aware of this. They're using a particular type of stem cell for which the preclinical evidence is lacking. So these are not strict embryonic stem cells, they're not induced pluripotent stem cells, they're a particular type of stem cell which is said to be ethically less contentious, but scientifically very under-researched. Their preclinical data on which this trial is predicated shows this is a surviving cell, so they have transplanted in their cells. This is the best picture they have of a surviving transplanted cell. Remember, you're aiming for at least 100,000 to get any clinical effect, so 1 is a long way from 100,000. If you look at the clinical efficacy in their preclinical non-human primate studies, you can see that the control here, so these are the animals they've been lesioned, the control is they've had nothing. This is a low dose and this is a high dose. There are no error bars on here, so we don't know how consistent it is, but the general trend is that just leaving the animals to do nothing is as effective as transplanting these cells. So this is a concern because they are transplanting 30 million of these cells into people's brains when the preclinical evidence that they will work is lacking. That led us to write a commentary in the Journal of Parkinson's Disease about this trial and the anxieties that it generates, because a failure of this trial or worse, a death in this trial will impact on the whole field of cell-based therapies and everything that everyone else is striving towards. So if you are thinking of being involved with this, and I can see why people would be tempted to it, might I refer you to the International Society for Stem Cell Research, who have an incredibly helpful website which patients-friendly to look at, which will instruct you as to whether you think these are trials worth doing. And for the clinicians here, or for the clinicians looking after the patients, ask your clinician to actually look at this website to make sure that they are happy taking forward any trials that they're asked to do. So to conclude, where have we got to with cell-based therapies over the last 35 years? Where are we in 2016? They've produced mixed results over the years, and that is as much to do with trial design as it is to the cells which we've actually tried to use as a therapeutic agent. What we can say with fetal dopamine cells is when it works extremely well. So this is a highly competitive, effective therapy, but it doesn't always work well. And that is a problem that we have to solve. But going forward, using stem cells, we can be much more prescriptive about the cells that we generate of the type we want, and we can be cleverer about how we then trial them in patients, making sure that we pay attention to some of the things that have gone wrong in the past and make sure it doesn't happen again. But beware, and I must emphasize, beware of any new advertised stem cell trials, especially if you're being asked to pay to join it. This is experimental medicine. Experimental medicine should be funded by agencies working out whether this is a therapy worth having. It is not a therapy that you should be paying for to have trialed in your brain. And as I say, if we don't pay attention to this, and if we're not more vocal about this, we will end up in the situation that this whole field could disappear because of some ill-conceived, poorly put together trial with a cell that is potentially lethal in a way that I think does great disservice to all the many people who've put years of work into this field. And finally, I'd just like to thank all the people who make this happen. This is a huge effort to take these cells forward. It isn't me on my own. There is a vast number of people who help me with this. There is my lab, the Trans-Euro Consortium that has been a fantastic source for knowledge and moving forward in this whole area. G-Force PD, which has been a fantastic innovation. And finally, a few individuals who I just wanted to flag up. Danielle and Agneta, who work with me and with Marlin to take forward these new protocols. And finally, Marlin herself, this was the best picture I could get of her. This is us when we're in the regulatory agencies in London, because Marlin has had the foresight as a basic scientist to see that there is no point working on a science that stays within the lab, but understands that the science ultimately has to make a difference to patients, and has paid attention very early on to that necessarily translation, with the hope that in 2 or 3 years' time, in Kyoto in 2019, we can tell you that such trials have begun. Thank you very much.